Good evening and welcome to the free webinar entitled Performance Evaluation Plan and Report. My name is Sylvia Angel, Senior Consultant at Medide, where I lead the IVD activities. Medide is a company specialized in quality, regulatory, and clinical activities related to medical devices and in vitro diagnostic devices. Today, Julianne and myself aim to provide you useful insights on how to prepare for compliance to IVDR in relation to the clinical evidence and particularly with the performance evaluation plan and report. Julianne has an extensive experience with questions related to the clinical activities and IVD, preparation for the IVDR transition, and setting up strategies for compliance. During the webinar, we will ask you a few questions that you will see appearing at the right side of your screen. Do not hesitate to answer. If you have questions during the presentation, you can directly write them in the chat screen, which is also at the right side of your screen. We will answer at the end of the talk. For those questions that will not be answered because of lack of time, you can always send a mail to Julianne or myself after the webinar. Julianne, the floor is yours.
Thank you very much, Sylvia, for this introduction. So indeed, good evening, everyone, and welcome to this webinar. So I think as a first important message, or just to set a bit the ground, a message that you are already all well aware of is that we will quite soon have to switch from the in vitro diagnostic directive to the in vitro diagnostic regulation. And hence, as a manufacturer of IVD medical devices, we will need to fulfill with requirements of the new regulation. So there is a lot to prepare for this transition, and one of these being in relation with the performance evaluation of an IVD medical device. So indeed, the prior, under the IVDD, a legal manufacturer of an IVD medical device was obliged to meet the essential requirements as per Annex 1 of the directive. And now there was a change towards general safety and performance requirements, which are listed in Annex 1 of the regulation. Specifically now, in relation with the topic today, there are some GSPRs which are tightly linked to performance evaluation of an IVD medical device. These are the GSPR 1, 8, and 9, which need to be where the manufacturer needs to demonstrate compliance with these GSPRs, meaning that they need to show that an IVD achieves its intended purpose, that it is safe and effective, that it is shows an acceptable risk-benefit ratio, that foreseeable risks and undesirable effects are minimized, and that the device achieves its analytical and clinical performance. All of this under the performance evaluation already needed to be demonstrated under the directive, but now comes as well an additional aspect to this. It's that it needs to be formally written or reported in a performance evaluation report, and this will be part of the device's technical documentation. So this performance evaluation, so it's to demonstrate illustrate the clinical evidence of an IVD medical device. So this clinical evidence is there to show that the device is performing as intended, and that it has clinical benefits, and that it is safe to be used. And this always with regard to the state of the art in the medicine. So it's always important to really compare the device that is being evaluated with regard to what is already known as part of the device's state of the art, and always to put it in relation with the state of the art. So this clinical evidence, so as already mentioned before, it needs to demonstrate that the device meets the relevant general safety and performance requirements. so the GSPR 1, 8, and 9. And it is then well illustrated as part of the technical documentation, where under the IVDR Annex 2, it is stipulated that the technical documentation needs to contain a performance evaluation plan, and this for all IVD medical devices, no exception at this level. And as well, it needs to contain a performance evaluation report or PER. So what is now performance evaluation for a medical IVD device? So it is composed, as illustrated here, I would say, of 4 different pieces, like a puzzle that fits together, where first it addresses the scientific validity, the analytical performance of the device, the clinical performance, and then also takes into consideration the post-market performance follow-up activities performed on the device itself. Now, first, a little bit more to the scientific validity, which is one piece of this puzzle. And when speaking about the scientific validity, here it's important to understand what this actually means. So here, the device itself does not really enter into the equation. It's more the intended purpose of the device. So basically, which analyte is going to be assessed and for which purpose. So the association of an analyte with a clinical condition or a physiological state needs to be demonstrated. As an example here, when we take, for example, anemia, it is well known that anemia can be assessed with a drop in hemoglobin levels. So here the scientific validity would be to demonstrate or to show that indeed there is a link between the level of hemoglobin and the diagnosis of or treatment efficiency of anemia. Same, for example, with the C-reactive protein, which would be used as an analyte, for example, and here different indeed clinical conditions could be associated with this marker. It could be like linked with an infection, inflammatory disorder, risk, prediction of cardiac disease, depending on the intended purpose of the device. Now, for well-known analytes, it's not essential to really, you need to demonstrate it through a brief rationale that there is a connection between the analyte assessed and the medical clinical condition, but it can be in a brief rationale based on information from textbooks, peer-reviewed literature, etc. It is a bit more tricky when the analyte is, or the intended use of the analyte is new. For example, a new biomarker for cancer, and there the scientific validity needs to be demonstrated clearly, and this, for example, through information on the scientific validity of devices measuring the same analyte or marker, through scientific peer-reviewed literature, through consensus expert opinions positioned from relevant professional associations, from results from proof of concept studies, or from results from clinical performance studies. So then, depending on where you stand as part of the literature reviewed, you might need to do indeed a small proof of concept study in order to really clearly demonstrate your scientific validity. And this scientific validity needs to be formally written down or formalized in a scientific validity report as per requirements of the regulation. Now, there are various tests in the world of IVD medical devices that can be categorized as established and standardized tests, or established non-standardized tests, news tests, and then is also the IVD classification that comes into consideration here. Now, scientific validity, however, needs to be demonstrated for all of these IVD medical devices in the same way, and this as well, not depending on which classification, IVD classification. So scientific validity needs to be demonstrated for all IVD medical devices. Then when it comes to the second part of the puzzle, which is the analytical performance of the device, there it refers to the ability of the device to correctly detect or measure a particular analyte. So here really the design of your device comes into place, because you need to demonstrate that you are able with your device to correctly assess the analyte. And there are, as per requirement of the IVDR Annex 1 and as well Annex 2, several aspects that need to be considered as part of this analytical performance demonstration, which are accuracy of the device, precision, analytical sensitivity, as well as analytical specificity, the limit of detections and quantitation, measuring range and linearity, non-linearity, the assay cutoff, as well as appropriate criteria for specimen collection and handling and control of known relevant endogenous and exogenous interference and cross reactions. So these are tests that need to be performed with the device in order to demonstrate its specific analytical performance. There are some guidelines which have been published to help guide in how to design such testing on devices, which are like the common specifications, which will be published more and more now as part of the In Vitro the IVDR. There are the CLSI standards, also the ISO standards, and other like relevant guidance, for example, from the WHO. So analytical performance of the device needs to be demonstrated through testing performed on the device, and then these studies, they need a result of these studies, they need to be documented in an analytical performance report. When we come here a bit to the same table illustrated before, when now talking about the analytical performance of the device, this as well needs to be equally addressed, whether it is an established and standardized test or a non-standardized test, or whether it's a new test. And it also is not depending specifically on the IVD classification. So analytical performance is also something that needs to be demonstrated for all devices. Then we come to the third part of this puzzle, which is the clinical performance. We have already talked a bit about clinical performance and clinical performance study last week. So here on the clinical performance, what needs to be demonstrated is the ability of a device to yield results that are correlated with a particular clinical or physiological or pathological process or state in accordance with the target population and intended user. So as part of this clinical performance, several aspects need to be demonstrated through clinical data. And these are, for example, the diagnostic sensitivity, so the true positive rate of a device, diagnostic specificity, the positive predictive value, the negative predictive value, the likelihood ratio, and the expected value in normal and affected population. So these are all aspects are to be considered as part of a demonstration of the clinical performance of an IVD medical device. And these now are no longer assessed with, needs to be assessed within a clinical setting. So using a clinical sample as relevant to the intended use of the device. So this clinical performance, it has to be proved based on data collected from clinical performance studies. So we've discussed this already last week. So based on the outcome of a performance evaluation, you will determine if yes or no you need to perform additional clinical performance studies in order to reinforce certain aspects of your clinical performance, or if you have sufficient data actually to waiver an additional clinical performance study on your device. It can also, in certain circumstances, be based on peer-reviewed literature, and this literature which identified studies which were performed with the device under evaluation, as well as in certain circumstances, it can also consider devices predicate devices. We will talk about this a little bit later. And it also can be based on published experience gained by routine diagnostic testing, which, of course, for legacy devices is also an important source to be considered. So clinical performance studies, they need to be performed unless you can duly justify that it is not needed because you have already sufficient clinical performance data. And all of the clinical data, they need to be compiled within a clinical performance report. So here again, when we come to the same table as above, we can see here that now there is a little bit a difference depending on the type of IVD device we are working with. So for well-established standardized test, which could be, for example, a glucometer, which is used within the context of diabetes. So meaning that this device it's established, it's already for a long time on the market, and it's a standardized test because between two identical or devices from different manufacturers, the result obtained with the device will always give the same an absolute kind of value. So for these type of tests, the clinical performance of the device can be based on other sources than clinical performance study performed by the manufacturer, but it can be based on findings coming from the literature. And so, for example, if you make a search on a specific glucometer coming from another manufacturer and you find there a study with relevant clinical data, this clinical data can typically apply to your device as well, and you can base your clinical performance on this data. For established and non-standardized tests, so here these would be tests which are already on the market where there are predicate devices, but where the outcome of the test might slightly differ from one manufacturer to the other because it's, for example, based on an antibody which might have a higher or a different sensitivity to the analyte. For such type of devices, it is also possible to base your clinical performance on the literature. However, you need to have priorly established a method of comparison between your two devices. So as an example, you have a predicate device on the market, you make a back-to-back comparison in terms of analytical performance, you assess that the two devices perform at the analytical level that they perform like in the same way. Then you could use data which are published on the predicate device. You could use this clinical data to support the clinical performance of your device under evaluation. Now for new tests, this is then a bit different because their clinical performance study are required because there are no other proof for clinical performance yet published. So these new tests will need either prospective or retrospective clinical data now for transition towards the IVDR, and most likely prospective because as it's not placed on the market, retrospective data will not be there. Then here, in terms of IVD classification as well, the level, however, of this clinical performance that you need to show will differ a bit depending on the risk class. So, of course, with devices which are under Class D, they will need a higher level of clinical performance evidence than Class C devices and Class B or Class B devices. Then for Class A devices, which are usually instruments or so used in laboratory setting, there is no clear clinical performance associated with the device. It's more going to be the aspects of performance which are more technical and not really of clinical relevance. Then fourth piece of this puzzle would now afterwards, once the device has been CE marked and is placed on the market would be the post-market follow-up data that you will collect on your device. So as the post-market performance follow-up, so PMPF studies or PMPF data, this is a continuous process that updates the performance evaluation of the device. It is there to proactively collect and evaluate performance and relevant scientific data with the aim to confirm the safety and performance of the device once it's placed on the market, to identify previously unknown risks, to identify emergent risks, to ensure the continuous acceptability of the benefit-risk ratio, as well as to identify possible systematic misuse of the device. So as a manufacturer, you are obliged to have or to perform such post-market performance follow-up activities unless you can really clearly, duly justify that these are not required. But these performance PMPF activities, they will then be consolidated in a PMPF evaluation report, and this data will then afterwards as well feed back into the performance evaluation report and consolidate the performance evaluation of the device on a continuous basis, so throughout the life cycle of the device. Now here a bit to summarize all of this. So performance evaluation is always done according to a performance evaluation plan, which can be established already very early in the process of design and development of the IVD medical device. This performance evaluation plan, it contains information such as the device description, the like intended use of the device, the target population, the safety and performance claims made by the manufacturer, the clinical benefit of the device, what type of applicable standards or common specifications exist for this type of device. It sets the clinical background and the state of the art. So this is really important because you will always have to measure your performance in relation or to assess performance in relation to the state of the art. And it will also contain information with regard to the PMPF plan that has been drafted for the device. So a plan needs to be established. It's also continuously can be updated throughout the life cycle of the device. Then based on this plan, you some activities also need to be performed, which are related to the scientific validation of the scientific validity of the device, the analytical performance of the device, as well as the clinical performance of the device. And all of this information is then compiled within a performance evaluation report, which once the device is placed on the market and there is additional PMPF data, will also be included into this report. And aim of this report is to demonstrate on a continuous basis that the clinical benefits of the device are obtained or achieved, and that when the device is used as intended, the benefits associated with use of the device, they outweigh the risks associated with use of the device. So this is a process, as now mentioned, but I think it's really important to emphasize this. This can be initiated already very early in the process of the design development of the device, and it can continuously be updated. It's something that you need to take care of on a continuous basis, because you need to, for example, if there is an additional risk that you have identified, which is associated with use of the device, this needs to be as well taken into consideration in the entire performance evaluation, and to afterwards reassess if there has been a change based now on the benefit-risk ratio. So it's a continuous process. It's not once it's done, it's frozen in time and you never go back to it. It needs to be updated on a continuous basis. And it's going to be most certainly one aspect where notified body will focus their attention on, because it's something that's currently for legacy devices they have not maybe assessed in such with such scrutiny before. And especially when you consider that a lot of devices were self-certified under the directive, and now most of the devices will require a notified body. One can expect that the notified body will assess performance evaluation of the devices. So it's important to be prepared and to structure this documentation well, as to have a good technical documentation ready for CE certification. So thank you very much for your attention. I hope you were attentive and for participating in this webinar, and we are ready to as well take your questions if there were any.
Thank you, Julianne. So first of all, I will answer to one question, which is the webinar recorded so that one can access it again at a later time? So the answer is yes, the webinar is recorded, and you will receive, because you attended to this webinar, you will receive a mail containing the link to the record and also, I think, the slides in the PDF format. Additionally to that, I see a question here. How frequent should PMPF related activities be carried out?
So this is a continuous process as well. It's established within a PMPF plan that you need to draft, and the activities performed, the PMPF activities performed, they need to be justified based on the performance evaluation of your device. So depending on what are the already available data for your device, you will need to, or is the device really, the safety and performance really well established? Maybe you will be able to justify a smaller amount of PMPF activities, but it needs to be seen as a continuous process as well. And then it also should justify, for example, the frequency of writing a PMPF report, and as well the update actually of the performance evaluation report needs to be justified. So the frequency at which you update such a report. So this is then, as you need to justify these activities clearly based on the level of clinical evidence that you have. And then it's to be seen as a continuous process. So
it can also be linked to the post-market surveillance, right? And feedback that you have from the field. One question, in fact, is linked to is it possible to give an example of frequent misuse that can be assessed during the PMPF? Would you have an idea of an example related to this?
So this would be, for example, if as a legal manufacturer, you realize that within a laboratory setting, a device is used, for example, to, to assess the a specific analyte, but it's used to diagnose, for example, a disease which you have not yet included as part of your intended purpose, and for which you have not brought the clinical evidence that it actually works for this, for example, type of diagnosis. This would be a systematic misuse of the device, which, clearly use of the device for this purpose would not be justified, as there is within the hands of the manufacturer no clinical evidence to support such a use. So yeah, that would be a typical misuse. Don't know if you have another example, Sylvia?
No, I think that your example is correct. Or if we, as a manufacturer during the post-market surveillance of the device, you are aware of some systematic use, typically, for instance, a device which is intended to be used with plasma, and systematically you see users which are trying it with full blood or with serum. This is something that you may assess throughout the PMPF. Also, we have to link it to the risk assessment, and we have to keep in mind that risk assessment is closely linked to the PMPF activity. So if you have, again, data which shows from your post-market surveillance that there are some risks related to the use of the device because of some misuse or additional risks, those should be into the risk assessment and should be reflected in your post-performance evaluation report as well. One question is also related, is there any format requirement for PEP and PER described in the IVDR, or must only the content be fully covered? Can we, for instance, work with an Excel file containing the correct information?
There is no clear format.
Template,
template or so for performance evaluation report or plan. It indeed, the IVDR provides the content that needs to be within these documents. Now, I would say that I have never seen, I must say, a performance evaluation plan and report in the format of an Excel sheet. It's rather when we do performance evaluation, it's more that you assess, for example, through systematic literature review, you would identify potential sources for clinical evidence or literature that you identified through a search, for example, on PubMed or on Embase. And then we use Excel in order to assess the relevance of these articles, so to exclude them, for example, at the title level, at the abstract level, and then at the full text level. And we usually document this within an Excel sheet that we attach to the performance evaluation report. I have never seen actually a real performance evaluation report in the form of Excel document, but it's not that it's stipulated somewhere within the regulation on the format that needs to be used to compile all of this data. But I would, however, still recommend a more classical Word format.
I think that in Excel file management of the document will be quite complicated, especially with the link that you have to do to make to all the references which are going through your PEP. It will be, I think, a nightmare in an Excel file.
Yes, as well, in a performance evaluation report, it's always nice as well to illustrate the state of the art, sometimes with a figure and so on. And this is going to be a bit more troublesome using Excel indeed.
We have one question in the analytical parameters. How to measure analytical parameters when no certified reference material exists? So I will try to take this one, Julian. So indeed, for some analytes, there's no such a certified reference. So then we have to find another way of measuring and assessing the analytical performance of the device. So one way to go is to use spiked sample, meaning that you spike samples, such as if your device is intended for serum, you will spike serum samples which should be negative for your analyte, with no concentrations of the known concentrations of the analyte, which is purified, if possible, purified analyte, or you measure the analyte by another method. Ideally, what I was doing as in my own experience is to let's say, try to analyze the samples which are used throughout the analytical performance by a certified lab, in fact. So the concentration of the analyte is measured by a laboratory who is certified. One question which also came, do we need to include usability studies in the PR, Julian?
So this is an interesting question indeed, because it's not something which is really clearly stipulated within the regulation. However, usability data are gathered and are important as well to demonstrate that when used as intended by the intended user, the device is performant and it is safe. So it is, for me, clearly part of the performance evaluation of an IVD medical device, as much as it is for a medical device. And as such, has its place in the performance evaluation report as well. So it is then a bit difficult to structure exactly where we're going to place this data because it's not formally part of the scientific validity, nor is it part really of the analytical performance or the clinical performance. But I suggest adding another section which talks about the usability data to demonstrate that relevant testing has been performed to demonstrate that when used as intended by the intended user, the device is safe and performant.
Thank you, Julian. This is also is very important for self-tests and also point-of-care tests, which now have related GSPR requirements related to the usability. Well, the way they are used in the current practice. So this is an important setting, in fact, for the self-test and point-of-care. What about devices which are on the market for more than 20 years, for which are widely used in the medical practice, and maybe the manufacturer, as the devices are on the market for so long, they do not have clinical performance studies available or usability studies? What would be the best approach in order to prepare to the IVDR transition?
Yes, so let me maybe go back a few slides here. So I think here this table here summarizes quite well the situation, because if you have a device which is a legacy device which has been on the market for more than 20 years, most probably it will fit into one of these categories. So either it's an established, so this for sure, but either standardized test or non-standardized test. And there it will depend indeed on the device itself. But as mentioned before, for such devices, I mean, there is no clear guidance yet really with relation to the IVDR, but what is under the GHTF guidances, as well as now published by MedTech Conference, it says that you can base your clinical performance on a predicate data from a predicate device. So in case it is a standardized test, you assume already that the results between the two tests are exactly the same. So you can take the data from the predicate in order to substantiate the clinical performance of your device. And if it's a non-standardized test, you need to have a method of comparison between the two devices in order to, prior to using the clinical data of the predicate device, demonstrate that they perform in terms of analytical performance the same.
Is it possible to use post-market surveillance data on that?
So yes, indeed, afterwards. So this is the specifically stipulated now for medical devices. It's the NDCG guidance 2020-06, which says that for legacy devices, you can clearly lean on collected PMS data to demonstrate the performance and safety of your device. So as long as there was a quality management system in place which allowed the correct collection and assessment of this data throughout these years. So it is indeed important for these type of devices to use the PMS data as well in order to support the performance of your device, to demonstrate that there have been no complaints, for example, with regard to the expected performance of the device, that there have not been any incidents related to the use of the device, etc. So this is then going to be as well a critical source to demonstrate that your device is performant and safe for when used as intended.
Another point that we have with in vitro diagnostic devices, which we do not have with medical devices, are those external quality assessment schemes, where you can enroll in one of those external control schemes and you participate. So it means that a few times per year, probably 6 times per year, you will receive a blind sample that you will have to assess and send to the scheme, send your result, and your result will be assessed and compared to all the other participants. So also this can be included, right, Giuliani, in the PR as an example of performance and also safety in a way, no?
Yes, exactly, especially if you have a predicate device indeed for which you have good clinical evidence, and then you do these, you participate in such testing, you can demonstrate on a continuous basis that you perform at the level of analytical performance as these predicate devices, so that yeah, there is no difference at this level. So as with regard to what is there already on the market and used as part of state-of-the-art good practice within like a good practice, there is no like big deviation between performance of your device and performance of the other predicate device.
Going back to the clinical studies, one question is there a threshold or standard explaining how many subjects should be included in a clinical study?
So this is a bit different than, for example, for analytical performance studies, where there are some guidelines which help you actually design these studies in a sense of how many samples you need, how often do you need to repeat this testing. For clinical performance studies, it's difficult to actually publish such guidances, because it will really depend on your medical IVD device, as well as, I mean, intended use of the device. Is it now, for example, used for screening a rare disease? Is it used for diagnostic purpose, or is it used for a diagnosis in combination with other means, for example, so only as a support in diagnosis? So all of this will then actually allow you to determine what is the sample size, clinical sample size, which you would need in order to have clinically relevant results, which are statistically significant. So you always need to take into consideration various aspects to design a study which is powered and which will lead to hopefully statistically significant results.
Thank you, Julian. I think I will take a last question. When are common specifications expected to be published for IVDs? So I believe that this is related for for Class D devices.
So currently, we hope that there are going to be, more and more publications of common specification for Class D IVD devices. To be expected in order of timeline. I don't know, Sylvia, if you have a bit more insight into when we can expect more of these, but they should come out on a regular basis now in preparation to the transition as well. But in the meantime,
Sorry. So what we know for the moment, in fact, we have, common specification only exists for Class D devices and not for all the devices. For the moment, under the IVDR, we only have the common specification for COVID test, which will be classified as Class D devices. However, do not forget that under the IVDD, so the directive, there are already some devices which are classified as list A. So I'm talking about the HIV, hepatitis diagnostic tests. Those devices will also, they have under the IVDD common technical specification. So what the European Commission is saying is that most likely all those devices which already have common technical specifications under the IVDD will be transferred as common specifications under the IVDR. So hopefully for people who are already working, for instance, on a device like, for example, transfusion, which is intended for blood grouping during transition, which is already a Class A under the IVDD, common technical specification exists. So if you are working and developing such a test, you can use the common technical specifications which already exist. For the other ones, well, we know that in the upcoming year we should have, but well, for the moment we cannot say much. We don't have a crystal ball. Okay, so thank you very much for your participation. I hope that you enjoy our webinar. So again, the presentation will be available. In fact, you will receive a mail from our side with all the information, with a link to the webinar, which was recorded, and also to the slides. Do not hesitate if you want to learn more about Medidate or the training that we are providing. Do not hesitate to go on our website. You will find all the information over there. Thank you very much for your attention and have a nice evening.
Thank you very much. Bye-bye.